Good morning. Today's lecture will be focusing on the cell structure and the function. So in this lecture, at the beginning of the lecture, I'd like to focus on the cellular organization level. As in the previous lecture, also we have discussed about the some organization level of the cellular. And in there we have discussed main elements and the molecular compounds which is responsible for making the cellular structures, which are proteins, carbohydrate, lipids, and the nucleic acid. And in this lecture, we are a little bit getting in a higher level, which we are going to discuss some organelles in the cell. So in here, in this lecture is focused on discussing about 10 different cellular organelles which is inside the cell and doing some various functions, as well as this lecture is focusing on the cell division as well as some signaling process of the cells as well, right? So here are the objectives of the lesson. At the end of the lecture, student will be able to understand the structure and the function of the cell organelles as well as the cellular processes such as cell division and the cell metabolism, as well as at the latter part of the lecture we'll be focusing on the cell communication and the signaling. So here is the structure of the cell. At the beginning, I'd like to focus on the cell membrane, which is creating the outer membrane or the margin of the cell. It is made by the cell membrane. And inside the cell membrane, we can see a huge cavity, which we called as the cytoplasm. So inside that cavity, there are different organelles are sink in there. So those are the things we are going to discuss further. What are those small apparatures which are sink inside the cytoplasm? The major one, which we can identify even in a light microscope, is called the nucleus, which is the largest organelle in the cell. Here, the purple color here, spherical shaped structure is the nucleus, and inside that we can see the nucleolus. The blue color thing is the nucleolus. The purple color huge one is the nucleus. We are going to take the nucleus out and discuss what are the different structure, how it made, as well as some function of the nucleus. As well as here you can see the cytoplasm, and in the cytoplasm you can see this endoplasmic reticulum, which is creating a tubular structure throughout the cytoplasm, which is responsible for transportation. And the mitochondria, the sausage-shaped structure, which are the powerhouse of the cell, which provide the energy or the ATP to the cell, is done by this mitochondria. And the Golgi apparatus, which are responsible for packaging, modifying, and transportation of proteins, lipids, we are going to discuss further. And another important organelle is centrioles, which is really important during the cell division process. And these blue color tiny vesicles are called lysosomes, which carry various enzymes. And these tiny particles are called the ribosomes, which is spread throughout the cytoplasm. Okay, those are some main organelles which we are going to discuss further, taking one by one. Okay, now let's focus on the nucleus. As I previously said, the nucleus is the largest organelle in the cell, which we can even see using a light microscope. And in nucleus, actually, it's having double cell membrane. It is covered with the double cell membrane, which we call as nuclear envelope, right? That's the nuclear envelope. And inside the nucleus, we can see the nucleolus, right? This yellow color part is the nucleolus. This endoplasmic reticulum, something is not related to the nucleus, it's just raw because those are connected to each other, but it's not a part of the nucleus. So this nucleus is this purple color spherical substructure, right? So this envelope is having some pores which is responsible for the transportation actually. And here we have the nucleolus. So what is the main function of the nucleus? So inside the nucleus, the genetic materials are stored. What are those genetic materials? The DNA, if it is related to the animals, of course, it is DNA, because DNA is, if it is related to the human, because the DNA is the nucleic material that store the genetic information. As it store all the information, it is act as the center of the control in the cell. Okay, that is the controlling center of the cell. That is about the nucleus. And then another important organelle is a mitochondria. So in mitochondria, it is having a sausage shape, and it also have double membrane, the inner membrane and the outer membrane. The inner membrane is folded inside the cavity and increase the surface area. So what is the main function of the mitochondria? It is responsible for production of ATP during the cellular respiration. As we are doing the aerobic respirations, that mitochondria is take place, right? And it is calling as the powerhouse because it's producing ATP. So that is about the mitochondria. And the third cellular structure we are going to discuss is the ribosome. Ribosome is something a really tiny particles which is having another nucleic material which we called RNA. In the previous lecture we discussed there are two types of nucleic acids, DNA and RNA. DNA is something inside the nucleus. The RNA is in the cytoplasm. And there are several types of RNA, tRNA, mRNA, and rRNA basically. So the ribosome is consisting with rRNA. So what is the main function of the ribosome is it is the place where protein synthesis is taken place. We know that transcription and translation is occur during protein synthesis process, which we discussed in last lecture. So the translation is take place in the ribosome. And actually it is having 60S and 80S two subunits. And the protein, while production of protein, this mRNA chain is reading by these ribosomes. Okay, so it is basically responsible for production of proteins. That's the main function. And the fourth structure, fourth organelle is the endoplasmic reticulum, which is really important organelle in the cell because it is doing kind of a transportation of basic components throughout the cell. Depending on whether it is connected with the ribosome or not, we can identify main two types of endoplasmic reticulum. Normally, we are calling it as ER, endoplasmic reticulum. If the ribosome is connected to the endoplasmic reticulum, we call it as rough endoplasmic reticulum. In simply, we call it as RER. And if the endoplasmic reticulum is not connected with the ribosome, then it is smooth. So smooth endoplasmic reticulum, which we simply call it as SER. The main function of the RER and SER depending on their structure. If the RER is having the ribosome, the main function is from the ribosome they are synthesis protein. Those synthesized proteins are transported by the reticuloendoplasmic units. Then if it is not connected with the ribosome, Then the main function of the endoplasmic reticulum is the lipid synthesis, detoxification, and the calcium storage. So the SCR and RER having two different functions. Another important factor for the Golgi apparatus, another important organelle is Golgi apparatus. It is a series of flattened membranous sacs. Okay, so those are like plates are gathering together, and we call those sacs are Golgi apparatus, or other words, cystarian. The function of these Golgi apparatus are it modifies, it stores, as well as it package the proteins and lipids. What are the main function? It modify, it store, and it's package the proteins as well as the lipids. And after that, at the end, part of the Golgi apparatus are turning into vesicles, and those vesicles are transported throughout the cytoplasm. Then the relevant parts are taking these proteins and the lipids. So the delivery function also done by the Golgi apparatus. Another important organelle is the lysosome. So in lysosome, they are actually tiny, tiny vesicles. It is only having a one cellular membrane, the cell membrane. Inside the lysosome, it is having cleavage enzymes. Now think that if the cell is going to be lyse, or if the cell is going to damage, or if the cell is getting old, that cell should be lyse. So that lysis function is done by the enzyme which is stored in the lysosome, right? Then actually the hydrolytic enzymes are containing in this lysosome. The main function is the cell digestive. The cell digestion is the main function. Another important organelle in the cell is the peroxisome, which are also tiny vesicles, but those are not, those are also containing enzymes that as the name implies, it is peroxisome. They can produce hydrogen peroxide during the chemical reactions. Okay, that peroxisome is really important to breaking down fatty acids as well as amino acid and detoxify certain chemicals. So they also play a role in synthesis of lipid and bile acid in the liver. Another important organelle which is in the cell is the centrosome and the centrioles, which is really important during the process of cell division. We will discuss the process, how it acts during the cell division at the mid part of this lecture. Okay, because the centrioles are actually functioning in all the phases of the cell division, nuclear division, and cytoplasmic division. The centrosome is like this, they are making up with microtubules, and you can see the centrioles, and the whole structure is called as centrosome. The centrioles are like spindle shape, cylindrical shaped structures. They are called microtubules. So these microtubules helping to form spindle fibers, which is responsible for separating chromosomes during the mitosis as well as the meiosis. Those things we will discuss in later. What are mitosis, what are meiosis? Just remember that centrosomes and the centrioles are responsible for the cell division process. And another important structure is the cytoskeleton. Actually, it is kind of giving a support. It's like the skeleton system inside our body, because it's make the shape, it's make the structure, it's give the support to our body. Likewise, for the cells as well, there is a structure which is give the support to the cells, which we call the cytoskeleton. So how is made? Basically, the protein filaments and the microfilaments, intermediate filaments, and the microtubules are responsible to make the cytoskeleton. It's kind of giving a structure to the cell, or it places the organelles where it is, and it's supporting for transportation of vesicles here and there. So the cytoskeleton is kind of a supportal structure to the cell. So last but not least, one of the major important organelle is the cell membrane, because this cell membrane is in the outside of the cell as well as inside of the cell. If you remember, we have discussed the nucleus is having double layer, and the mitochondria also having double cell membrane, and the peroxisome is having one cell membrane, lysosome is having one cell membrane, endoplasmic reticulum also the same, because the cell membrane is in everywhere. So you have to go thoroughly with the structure of the cell membrane. There are lots of animation in the YouTube. Better to go with these animations as well. Then you can get a 3D image into your mind. First I will discuss, then you can go and check some structure of the cell membrane. The cell membrane is having lipid bilayer. It is having phospholipid layers, two phospholipid layer. In phospholipid, we also discussed that the structures of the phospholipid in the previous lecture. It is having a hydrophilic head and the hydrophobic tail. The hydrophobic tail is always inside the cell, and the hydrophilic head is exposed to the outer membrane, outer surface. And as it is having a lipid bilayer, there are some protein elements which are sink into the cell membrane. If it is go to in towards the cell membrane, in both layers, we call it as the integral proteins, because it is sink into two layers. And if it is only sink into a one cell layer in one membrane, we call it as peripheral protein. Okay, peripheral protein. Actually, what are those proteins do? They helps to, it's kind of an opening, or it's kind of creating as a door to go things inside and outside of the cell. Protein channels are present. Through that channels, the varying things going here and there inside the cell. And the outer layer, we can identify some lipids. Basically, these lipids are connecting with they are responsible for the communication. Basically, if these carbohydrates are connecting with the lipids, we call it as glycolipids, which means these carbohydrate tiny chains are go and connected to the phospholipid bilayer. Then it is a combination of the carbohydrate and the lipid. Because of that, we are calling it as glycolipid. And there are some carbohydrate molecules which are connected to the proteins. Then carbohydrate protein, we call it as glycoproteins. Even it is a glycolipid or a glycoprotein, those are responsible for the communication of the cell. Okay, that is the structure of the cell membrane. What are the structure? It is having a lipid bilayer, and there are protein integrated in the lipid bilayer. If it is fully integrated, we call it as integral protein. If it is partially insert, we call it as peripheral protein. What is the main function of the protein? It's controlling the things going inside and outside of the cell. And other one is carbohydrate apparatures. If the carbohydrate is connected to the proteins, we call them as glycoprotein. If it is connected to the lipid, we call it as glycolipid, because they are responsible for the cell signaling. Okay, those are the main structure of the cell. The cellular organelles are in there. Now we are coming into one of the most important part of the lecture, the cell division, the second part of the lecture. Okay, the cell division. So why need to be happen the cell division? Because at the beginning we have the egg and the sperm, and we are having the oocyte. The first cell of our body is called as oocyte. That oocyte is divided again and again and making billions of cells and creating our whole body. So if the cell division is not taken place, this is never gonna be happen. So cell division is required for this growth, for the repairing mechanism, as well as for the reproduction. So, cell division is a fundamental process by which single cell is divided into two or more cells. The cell division, there are main two types of cell division processes. The one is mitosis, the second one is the meiosis. The cell division means simply the mother cell is dividing into daughter cells. Okay? During the process of mitosis, the mother cell is dividing into two identical daughter cells. In meiosis, at the end of the process, it creating four daughter cells, but those are not identical to each other. You will understand the differences between mitosis and the meiosis while we are discussing it later. Okay. Before going into the nuclear division, I'd like to focus on the whole cell cycle. The cell cycle is having main four phases. The overall whole cell cycle. This G1, S, G2 and the M. Here the G1, S, G2 is called as interphase. G1, G2, the all together is called as interphase. This M is responsible for the nuclear division as well as the cell membrane division. So G1 is the growth, S is the DNA synthesis part, G2 again growth, means the mitosis or the meiosis, and the cytokinesis is also taken place, which means the cell membrane division also happening in there. What you need to understand in the cell cycle, there are main four phases: G1, S, G2 and M. Out of that four phases, this G1 is G2 is called as interphase, and M is having the cell division. The cell division is happening in the phase of M. As I said earlier, there are two types of cells, cell division. First one process is the mitosis, the another process is the meiosis. In the mitosis, what happened? From the mother cells, at the end of the process, it's going to create two identical daughter cells. And in meiosis, it is creating four daughter cells at the end of the process, but those are not identical to each other. So we will discuss the mitosis and the meiosis one by one. Okay. The mitosis is a process which a single eukaryotic cell divides and produces two genetically identical daughter cells. It is happening basically in eukaryotes because there should be a nucleus there. That's why. So the mitosis is a crucial for the cell growth, cell development, as well as tissue repairing mechanisms. So there are several stages of the mitosis. The stages of mitosis. This mitosis and the meiosis is in the M phase. This interphase is something not related to the cell division part. It is just the stages of the cell cycle. It is G1, S, G2. It is just growing phase. The division phase is taken in M. That M is, we can identify two processes. One is mitosis, the other process is meiosis. Even in here, they have indicated the interphase as the stage of mitosis. It is not technically a part of the cell division because it is kind of a cell growing part. Okay, interphase, as well as in the picture which we've previously seen, it is having G1, S and G2. And when coming to the mitosis, the first stage is the prophase. Here you can see the mitosis. There are several stages. The first stage is prophase. Actually, this pro-metaphase is something intermediate phase between the prophase and the metaphase. So we are going to ignore that. There are main four phases. In the mitosis, there are main four phases: prophase, metaphase, anaphase, and the telophase. Those are the main four phases because this pro-metaphase, something is in between the prophase and metaphase, which we are not going to be discussed. But you need to remember, in the mitosis process, there are four stages: prophase, metaphase, anaphase, and the telophase. So first we will discuss what happened in the prophase. As you know earlier, that all the genetic materials are stored in the nucleus, right? In order to divide this cell into another two types of daughter cells, what should be happen? This nucleus also need to be divided. So first step is the nuclear membrane, or in other words, the nuclear envelope or the double layer of the nuclear membrane is gonna be dissolved. After that, all the genetic materials are come into the cytoplasm. Those genetic materials are called as the chromosomes. Those two chromosomes are connecting each other by the place called centromere. So here is the example for centromere. I'll show you the video, then it will be easily can find what is the centromere here. Yeah. You can see these are the chromosomes. How it happens? The nuclear membrane dissolved and all the genetic materials come into this cytoplasm. After it's come to the cytoplasm, it is getting condensed and it's clearly visible. It's gonna be visible. And the other thing that you have to remember is these chromosomes are connecting each other by the place which we call centromere. This is the place the centromere take place. This is the centromere of each chromosome. This is a chromosome. That chromosome having two arms. This is one chromatoid and this is another chromatoid. These two chromatoids are connecting by the centromere. The whole structure is called the chromosome. Inside the chromosome, the genetic materials, the DNA, is situated. Right. So that is what happened in the prophase. What are the things happening? The nuclear membrane is going to dissolve and the genetic materials are getting condensed and these materials are creating chromosomes. Those chromosomes are connecting each other by centromere. And after that, the centrioles, if you remember, the centrioles are made up with microtubules. Those centrioles go into the poles of the cell, north pole and the south pole, right? And then from those centrioles, they are spreading their microtubules towards the cytoplasm. And then those microtubules go and connected to the centromeres, which are in the chromosomes. So that's what happened in the prophase and the metaphase. So the second stage of the mitosis is the metaphase. In metaphase, what happened? These chromosomes are now in different places of the cell. Those things should be taken into the equator of the cell. The process of these chromosomes get into the equatorial plate is the metaphase. How it happens? Because centromeres are connected with the microfilaments now. Those are, one part is getting elongated, other part is getting shortened, and however, those are creating these into a one single layer, which is in the equator. For example is like this. You can see this is the equatorial plate in the cell, and these are aligned into a line by the centrosomes, centrioles. Okay. And during the third phase, which is anaphase, what they are doing, now the microtubules which is connected to the centromeres are getting shortened. Because of that pulling, these chromosomes are getting breakdown. The chromatoids are heading towards the poles because of the function of microtubules. That's what happened in the anaphase. What happens? The centromere getting dissolved and the chromatoids are heading towards the poles because of the action of microtubules. Now we have two genetic materials in two poles of the cell. Then what happened? Another new nuclear membrane is gonna make by covering those newly synthesized genetic materials. So that is what happened in telophase. Hope it is clear. You will get more idea when I'm showing you another video. So it is clearly explained the 4 stages of the mitosis. The integral phase is something not related to the cell division. It is happening in the cell growth. G1 is G2, something not responsible for cell division. The cell division is taken place only in the M. There are two types, mitosis and the meiosis. The mitosis also having 4 phases, meiosis also having 2 phases. We will discuss it one by one later. The 4 phases of mitosis are prophase, metaphase, anaphase, and the phase. So here are the structure. And before go to the meiosis, like to show you some YouTube video, then you can get the idea about the process of the mitosis. Hope you are getting a clear idea about the mitosis process. Okay, another process which cell division takes place is via the meiosis. Basically, the mitosis, the previous one that we discussed, mitosis is happening all the cells in the body. But meiosis is happening only specific cells in the body. We are calling them as gametes. What are those gametes? Sperm and the eggs, which are responsible for the reproduction procedure. So the mother cells or the germ cells are produce these sperms and eggs by the process of meiosis. We will discuss what is the process called as meiosis. The meiosis also having two stages, meiosis 1 and meiosis 2. Each of these stages is having the prophase, metaphase, anaphase, and the telophase. What are when coming to the meiosis, there are two stages, meiosis 1 and meiosis 2. Meiosis 1 again having prophase 1, metaphase 1, anaphase 1, and the telophase 1. Meiosis also having 4 stages. Meiosis 2 also having 4 stages. Those are prophase 2, metaphase 2, anaphase 2, and the telophase 2. After whole procedure is happened, it's gonna make 4 identical daughter cells. Actually, those are not identical. Those are going to make 4 daughter cells which are not genetically identical to each other. We will discuss what is the reason for that. Okay, so what you have to get an idea about the meiosis is it is happening only in the germ cells, which are eggs and sperms, and there are 2 stages, meiosis 1 and meiosis 2. Each of these stages also having anaphase, metaphase, prophase, and the telophase stages. Right. In the prophase 1, what happened? As it is same for the mitosis. Thus, chromosomes are getting condensed, and one specific thing is happening in here. The homologous chromosomes are pairing up. What I mean by homologous chromosomes? As we know, we are having 23 pair of chromosome. One set of 23 is coming from the mother's side. Another set of chromosome is coming from the father's side. So those 23 pairs are getting into a one place. Like similar chromosomes are getting close to each other. That is what we call homologous chromosome. Those homologous chromosomes are getting paired up. That is called synapsis. Okay, so that is happening in here. When they are getting closer to each other, like their 23 pairs is getting closer to each other, they change their genetic materials. That's why at the end of the process, it is not going to make identical daughter cells because the genetic materials are mixing in the prophase. Then what happened? The nucleus membrane is breaking down and the centrioles are going into the poles and they are making spindle fibers. In metaphase 1, what happened? These paired homologous chromosomes are aligned into the equator as previously discussed. Okay, it is exactly same to the mitosis metaphase. In metaphase, what happened? These homologous chromosomes are getting aligned into the equator. In anaphase, the spindle fibers are getting shortened and these chromosomes are heading towards the different poles. In telophase 1, what happened? Now we have 23, only a 123 set in either side. a set of 23 is in the North Pole, the another 23 of chromosomes in the South Pole. Likewise, after the cell membrane is occurred, now we have haploid cells. Haploid means it is only having half of the genetic materials, because at the end of the meiosis 1, we are going to have 23 chromosome cells, not 23 pairs. At the beginning, we had 23 pairs, but at the process of the meiosis 1, it is breaking down into 2 cells, but those 2 cells are having only half of the genetic materials, and those are not genetically identical. So that's what happened in meiosis 1. During the process of meiosis 2, now we are going to take a one cell which is completed the meiosis 1 stage, and what happened to that cell? Again, another mitosis is taken place. What happened in there? Prophase 2. Again, the nucleus membrane is getting dissolved. Spindles are heading to the North Pole and the South Pole. The microtubules are making spindle filamentous, and those are connected to the centromeres. And metaphase 2, what happened? These chromosomes are getting aligned into the equator or the metaphase plate. And then these chromosomes are again connected to the filamentous by the centromeres, and these filamentous getting shortened and creating the chromosomes are heading towards the poles. They are pulled to the opposite poles. And in telophase 2, what happened? These chromatids reach the poles, and again newly synthesized nuclear membrane is going to be synthesized at the end of the process. So at end of the whole process, stage 1 and stage 2 of the meiosis, we are going to have 4 daughter cells which are genetically differs to one another. So I'll show you another video which is describe the meiosis part as well.
In this lesson, we'll explore the details of what happens during the phases of meiosis. Meiosis, sometimes called reduction division, is the type of cell division that produces gametes. By gametes, we mean sex cells, such as sperm cells in males and egg cells in females. Meiosis is broken down into 2 stages of cell division, called meiosis 1 and meiosis 2. Meiosis 1 has 4 phases: prophase 1, metaphase 1, anaphase 1, and telophase 1. And meiosis 2 also has 4 phases: prophase 2, metaphase 2, anaphase 2, and telophase 2. Let's look at what happens during meiosis 1. Prophase 1 starts with a diploid cell. Its chromatin contains 2 uncoiled, spread out sets of chromosomes, 1 from each parent. After the DNA in the chromatin replicates, it condenses into the more familiar X-shaped chromosomes. The replicated DNA is the same in the identical sister chromatids of each chromosome. In a process called synapsis, each chromosome pairs up with and binds to its corresponding homologous chromosome, forming a tetrad. A tetrad is the group of 4 sister chromatids in paired homologous chromosomes. The chromosomes contain genetic information called genes. These genes were inherited from each parent. And different versions of the same gene on each chromosome are called alleles. In a process called crossing over, chromatids from each homologous chromosome exchange segments of alleles. Also called recombination, crossing over randomly happens on every chromosome, resulting in different gene combinations. This explains why every gamete is genetically different from every other gamete. Crossing over results in genetic variety in offspring. This is why children are different from their biological parents, as well as from their biological siblings. Continuing on with prophase 1, the nuclear membrane disappears. The centrioles move to opposite ends of the cell, and spindle fibers fan out from them. Next, in metaphase 1, the homologous chromosomes line up at the equator and attach to spindle fibers from opposite poles. During anaphase 1, spindle fibers separate the homologous chromosomes in each tetrad and pull them to opposite poles of the cell. The cell enters telophase 1 with one chromosome from each homologous pair at separate poles. However, each chromosome still consists of sister chromatids. Keep in mind that each chromosome's sister chromatids are no longer identical because of the allele exchange that happened during crossing over. Then, spindle fibers disappear and the nuclear membrane reforms around the chromosomes. Finally, cytokinesis occurs. Meiosis I ends with two genetically different haploid daughter cells. Each haploid cell contains only one set of chromosomes, consisting of paired sister chromatids. Both cells now enter the next stage, meiosis II. However, unlike meiosis I, DNA does not replicate before meiosis II begins. Once again, in prophase II, the nuclear membrane disappears, and spindle fibers fan out from the two sets of paired centrioles. During metaphase II, the chromosomes in each cell line up at the equator and attach to spindle fibers from both poles. During anaphase II, the sister chromatids of each chromosome separate and move to opposite poles. Once the sister chromatids separate, they're called chromosomes. Finally, during telophase II, the spindle fibers disappear and nuclear membranes reform. And cytokinesis occurs in both cells. Meiosis II ends with 4 genetically different haploid daughter cells, each containing only one set of chromosomes. Some key points to remember about meiosis. It begins with a diploid cell. Meiosis only produces gametes. Gametes are genetically different haploid cells, sperm cells in males and eggs in females. Meiosis has 2 stages of cell division, called meiosis I and meiosis II. During meiosis I, homologous chromosomes separate to produce 2 haploid cells, each containing chromosomes in the form of paired sister chromatids. In meiosis II, the sister chromatids separate in both cells, becoming individual chromosomes. Cytokinesis of these cells produces 4 genetically different haploid gametes. And here are some key points to remember about prophase I. The pairing of homologous chromosomes, called synapsis, occurs. Each pair of homologous chromosomes, consisting of 4 chromatids, is called a tetrad. During the process of crossing over, chromosomes in homologous pairs exchange segments of alleles. Crossing over results in genetic differences in gametes. All gametes produced by meiosis are haploid.
Hope you can get a clear idea about the meiosis and the mitosis part. So here, after you get the idea about the process of meiosis and the mitosis, you need to get the idea about how it differs, like how the mitosis differs from meiosis. The main one is the number of division. In mitosis, it is only a 1 division. And after the division, it is making two identical daughter cells. And those two daughter cells are diploid, which means each one is containing 23 pair of chromosomes. When go to the meiosis, there are two stages of division, meiosis 1 and meiosis 2, and it is resulting 4 daughter cells, and those 4 daughter cells are also genetically differs to each other. And coming to the chromosome number of years, mitosis, it's remained the same chromosome number as the parental cell. It is diploid. While meiosis, it reduced the chromosome number into the half. So we call it as haploid cells. So those are the basic differences between the meiosis and mitosis. There are more. You can further research, further read and get some idea about the differences between the meiosis and the mitosis. So at the first part of the lecture, we have focused on the cellular organelles, and the second part of the lecture, we have focused on the cell division procedure. And here, at the latter part of the lecture, we are focusing on the cell metabolism as well as the cellular signaling pathways. This cell metabolism is something that we have discussed earlier as well. In the previous lecture, we discussed what is metabolism means. Metabolism means the process of the combination of anabolism and the catabolism. Anabolism means making up, like small components are gathering together and make a huge structure. We call it as the anabolic process. Involve building larger molecules from a smaller one is called as anabolic process. How it happens? Like amino acids are gathering together and make a huge protein molecule. The monosaccharides are gathering together and making a polysaccharide. And nucleotides are gathering together and making DNA or RNA. Likewise, smaller units are gathering together and making a large one is called as anabolic procedure, and it requires the energy. When coming to the catabolism procedure, it is breaking down huge molecules into smaller units. In there, what happens? The energy is released. So here is a clear picture that gives you an idea about how the anabolic reaction and the catabolic reaction is taken place. In the anabolic reaction, the energy is required to produce the larger molecules from the smaller ones. In catabolic reaction, the larger molecules is breaking down to smaller molecules, while that the energy also getting released. Right. Examples for the catabolism, in the previous lecture also we discussed how the glucose is going to be catabolized and how the glycogen is going to be catabolized and the lactic acid fermentation. So all these things are clearly discussed in the previous lecture. You can go for that note as well to get an idea about that. And some example for the anabolism. And we discuss how the protein is synthesis, how the DNA is going to be replicated, and the glucogenesis and the glyconeogenesis. All these are coming to the category of the cell anabolism. Right. Some important factors about the cell metabolism: the energy production. Right? In metabolism, like metabolic pathways such as glycolysis, citric acid cycle, and oxidative phosphorylation produce ATP, which provide energy which is necessary for various cellular productions. So you know that during the catabolism process, it releases the energy. So that energy is required to do some cellular function. And the biosynthesis means these components are required for cell growth, cell maintenance, as well as the reproduction procedures. Another important factor about the metabolism is detoxification. How detoxification is happening? Because by the breaking down of the smaller unit. The large one is breaking down into smaller unit, and if there is something unnecessary for the body, we can remove that. So that is detoxification and the homeostasis. And if something is getting increased than the normal range of the body, then by the process of metabolism, we can again come into the normal range. So those are some important factors related to the cell metabolism. The last part of the lesson is the cell communication and signaling. Do you remember in the cellular membrane we have discussed there are some carbohydrates are connected to the lipid bilayer. If those carbohydrates are connected to the lipid, we call this as glycolipid. If these carbohydrate chains are connected to the protein, we call them as glycoprotein. Actually, those hanging carbohydrates are the ones which is responsible for mainly for the cell communication. Other than that, here are some types of cell signaling. Just remind the names. Insulin signaling, which I told you, we have discussed that in previous lecture as well. We discussed the pancreas. From the pancreas, it produces insulin, and those insulin is released to the bloodstream, and the insulin is going and connected to the cells, which are going to actually needed to absorb the glucose from the blood. So that is insulin signaling. Neurotransmitter signaling means for the neuromuscular junction, they release some neurotransmitters to the synaptic cleft, and then they are passing the signals from neurons to the muscles. That is happening via the chemical substances called neurotransmitters. And growth factor signaling, immune cell activation, and the calcium signaling all are some types of cell signaling process. Okay, at the end of the lecture, what we have discussed so far, in the morning, we have discussed the 10 cellular structures. We discussed the structure of the nucleus, structure of the mitochondria, Golgi apparatus, and the endoplasmic reticulum, and ribosomes, lysosomes, peroxisomes, and then the mitochondria and cell membrane. Likewise, we have discussed major cellular organelles. And the second part of the lecture is focusing on the cell division. In there, we have discussed the process of mitosis and the meiosis. And in the last part of the lecture, we have discussed the cell metabolism pathway. So here are the assignments. You can go and check with the presentation, and that's the end of the lecture. Thank you.